Assalamu alaikum. I am Shahriar Mohammad Shohan. My ID is 116150225. Today, my presentation topic is Human Health Risk Assessment. Under this topic, I will discuss about the analysis of dose and responses. First, we need to know what is Human Health Risk Assessment. Here is a general definition from EPS, Terms of Environment Glossary is that risk assessment is a qualitative and quantitative evaluation of the risk posed to human health or to the environment by actual or potential presence or use of specific pollutants. Now, dose analysis. When performing dose response analysis for health risk assessment, a clear concept of what type of dose has been used in the study that produced the available responses plotted in the dose response data must be specified. Here the term dose is used exclusively to describe any type of chemical exposure. This includes experimental dose as well as what is typically called external concentration or incidental exposure. Typically, animal studies utilize relatively high doses of chemicals so that unequivocal or clear observations of the effect can be obtained. These doses are usually much greater than the normal doses received by humans. Now, dose existing in the exposed organism. There are different types of dose that arise from scientific investigations, and these have different interpretations and should be distinguished properly while performing the risk assessment. There are primarily three types of dose that may exist in the exposed organism. First one is the administered dose or external dose. The administered or external dose denotes the amount of chemical administered to experimental animals or humans in a controlled environment setting by some specific route at some specific frequency or through incidental exposure in occupational or epidemiological studies. The second one is the absorbed or internal dose. The absorbed or internal dose is the amount of chemical that is systematically available and can be regarded as the fraction of the external dose that is absorbed and enters the general circulation. It is affected by the absorption, distribution, metabolism, and excretion of the chemical. The third one is the target and tissue dose. The target and tissue dose is the amount of chemical present in the specific tissue of interest. For example, total mercury or methyl mercury concentration in umbilical cord tissue. Now, dose calculation. Here are two formulas to determine the exposure and the intake dose. Now, response analysis. Toxicological, pharmacological and microbiological responses are generally related to an observation or effect seen following exposure. To discriminate or differentiate between these adverse and the non-adverse effects, considerations should be given to the observed effects. Now, the types of responses. There are 6 categories of responses in which most of the observed responses are included. First one is the continuous responses, quantal responses, counts responses, ordinal categorical responses, hierarchically nested responses, and multivariant responses. The continuous responses is this continuous response. First, continuous response. This measurement of responses is associated with each individual subject and can take on any value within a defined range of outcomes. For example, changes in the body weight. These types of data can be reported as either individual values or summary statistics. That is, for example, typically the arithmetic mean, measure of variability, arithmetic standard error or standard deviation, and the number of subjects per dose group. The second one is quantal responses. These are also called the binary or dichotomous responses. For each dose, the number of subjects responding out of the number of subjects available is recorded. Then the proportion of the subjects experiencing the event is reported. For example, the proportion of animals with a tumor in a cancer bioassay. Then count responses. It is expressed as a discrete number of items measured in a single experimental unit. For example, the number of papillomas on the skin. It could be 25,405 in women and 19,925 in men, which is seen in the figure. The ordinal categorical responses is that these generally take on one value from a small set of ordered values. Ordinal data are an intermediate type of data and reflect ordered severity categories. That is, they are qualitative data but with a rank order. For example, severity grades of the histopathology. Then hierarchically nested responses. Nested data or the clustered data occur when the observation is nested within a large group. Results from developmental toxicity studies are a common example of this type of data. Data are organized into like a tree structure. The last one is multivariate responses. These occur when multiple endpoints are measured for a single experimental unit, such as when genomic data consists of thousands of gene responses over different concentrations. Now, the factors affecting dose responses. These are the factors we can see in the figure which affect the dose responses after the exposure of the chemicals. Now, risk assessment decision. After a comprehensive analysis between the dose and responses, risk assessment decision can be made by two ways. First one is the Noel approach. Noel means no observed adverse effect level. In this approach is used when there is only one dose group and a control group, or when there is no adverse effect. And the second one is the BMD approach. BMD means benchmark dose. BMD approach mainly takes all of the dose response data into account and qualifies the uncertainty in the data. With this, I complete my presentation. Assalamu alaikum.